So in continuation to our previous video, we will see how an auto as well as an allo polyploidy are generated from a protoplast. So in case of an auto polyploidy, I think the diploid 2n number, we can make sure that the entire chromosomal set can be increased, the copy number can be increased from 2n to 3n. So it is called as a triploid, 4n, a tetraploid. So we can increase the whole process. So now why I am mentioning this 2n to 3n, principally because using this plant breeding, somatic cell fusion, and in also case of an auto polyploidy, so we also find in the market several seedless variety, especially with respect to the fruits like watermelon, grapes, papaya. So we have what you call this as a seedless variety. Now how this seedless variety is done is basically because of somatic fusion, where autoploidies are going to be found. Before we get into either an auto or an allo polyploidy, we will just discuss some of the key events which is going to happen in case of a cell division, and where we are going to do an interference. So in case of a cell division, I will not discuss the entire process. So now cell division can be studied under several stages. We have a prophase, we have a metaphase, we have a anaphase, and we have a telophase. Now this is a schematic representation only with one set of chromosome. Okay, so in case of an eukaryotes, we all know that we have a pair of chromosome, one from a paternal and one from a maternal. So in case of a meiotic cell division, usually we have a diploid 2n. Now this 2n is going to be reduced to n. There will be a reduction. Now the reduction is going to take place at two stages. There will be a first division followed by a second division. What happens in case of a very first division is that, so in case of the early meiotic cell division, these homologous chromosomes, they pair together. We want to make sure that the homologous chromosomes pair in that, and then we also discussed in the yeast vectors there is a homologous recombination. So now there will be a crossing over. Now the crossing over is a very important phenomenon, basically because during this meiotic cell division, the cell wants to make sure that we have the homologous chromosomes held together. So now these things are all there on a plane. Alright. Now what happens in case of the first division? Now these homologous chromosomes, after the homologous recombinations, will be segregated into two cells. Okay. So now what we call this as a normal segregation, in which a chromosome will be transferred to one cell and another chromosome will be transferred to another. This is called as a segregation. We also use another term called as a disjunction. Disjunction is nothing but a normal segregation. Alright. Now here what happens, these individual chromosomes undergo replication, because this replication will also start even in the prophase itself, in fact. So now when we do have the homologous chromosome, we have two copies of the same DNA sequence. Once these two copies undergo replications, so now we are going to have 4 copies of the same chromosome. So now the 4 copies which is going to be generated during the prophase and metaphase, what happens is also going to be segregated or disjunction into individual cells, in fact. Now that is the first division. Again this particular cells undergoes second set of division. Again they have the prophase, metaphase, anaphase, telophase from here. So what happens, I think these homologous chromosomes undergo replication. So this also undergo replication. In the second division, this will give rise to 2 gametes and this will give rise to 2 gametes. So now during this, then what happens individually, now here we have the 2n. So now here we have a single chromosome like this. So now this condition we call this as an N gametes. So now it is a 2N. So now by the meiotic cell division, so we are going to get this N. Alright. Now in case of a somatic cell fusion, what happens? So now usually we have a normal segregation and then we have a disjunction. Okay. So now we are going to create a condition what is called as a non-disjunction. So what exactly is a non-disjunction? In case of a non-disjunction, we don't want the segregation to happen perfectly. Alright. So now what are we going to do this? We are going to interfere chemically during the segregation, especially in case of a metaphase or an anaphase. So we are going to introduce an alkaloid, what we call this as a colchicine. So this colchicine is introduced in case of a meiotic cell division, either it is in metaphase stage or in case of an anaphase stage. So during this anaphase stage, what happens? The segregation takes place. So in case of a segregation, so once this cell will become two daughter cell, so the chromosomes has to be distributed equally between the two daughter cells. So now during this distribution, so usually what happens, we require what we call this as a spindle fibers. Spindle fibers do attach to the centromere, and then these spindle fibers will drag these chromosomes into individual daughter cells. How the whole process is going to be done? So the spindle fibers is made out of usually the tubulin. It's a protein. So usually the free tubulin, this is the monomeric units of the tubulin. So once we are going to have the formation of the spindle fibers, what happens? These free tubulin undergoes a process known as a polymerization. So those things are going to be joined together with the process of it. Then it will create a spindle fiber. So now it will bind to the centromere, these spindle fibers. Then what happens? This polymerized, so it is going to be a very long. So during the dragging, what happens? This spindle fiber will reduce their size. So what happens? This will undergo a process of what is called as a depolymerization. So now once the depolymerization starts, automatically, since it is binding to the centromere, this will be dragged to either side of the daughter cell, in fact. So we have a free tubulin, and during the formation of the spindle fibers, so then these free tubulins will undergo a polymerization, and then while dragging them, so we are going to have a depolymerization process. This is the protocol which is going to happen either in the early metaphase, and finally the segregation happens in the anaphase stage, in fact. Colchicine, what it does, it disrupts the spindle fiber formation. As a result, there won't be any spindle fibers attaching to the centromere of these homologous chromosomes, and there won't be equal distribution of chromosomes between the daughter cells. So in the presence of a colchicine, what happens? These chromosomes is unevenly distributed. Okay. Now we need a condition. So we will take an example, because in case of a cell division, meiotic cell division, there is a first division and there is a second division. This non-disjunction, it can happen either in the first division itself or in the second division itself. We will just see what happens if there is a non-disjunction happens in the second division. So we take, this is the normal cell. We have a homologous chromosome and we have 4 copies of chromosome. So in the very first division, it is going to be normal. So a pair of chromosomes is going to be distributed between the daughter cells. So now in the second division, there is a mistake. Non-disjunction process takes place. So now this daughter cell divides normally. So we are going to get the gametes with N and with N. However, this daughter cell, there is because of the colchicine interference, there is a non-disjunction because the spindle fibers are disrupted. So now this one is not equally distributed. So one cell will not get any chromosome, whereas the other cell do get an extra chromosome. So now here, in case of a normal, all the 4 gametes have N in number. But here, now this is N, this is N. So these 2 gametes are perfect. whereas the other two gametes, so we have this is an n minus 1, there won't be any chromosome, and we have this n plus 1. Okay, now you have created, so now there is a gamete with a full set of chromosome. Now there is a gamete which is a normal one with the reduced chromosome, and we have a gamete with no chromosome. Now assuming that this gamete fuses with this gamete, so now what happens, we are going to get a cell, a fusion cell, so in which instead of a diploid, so we are going to get a monoploid. Okay, in case of a monoploids, usually they don't survive, principally because usually we have a chromosome from one cell and another chromosome from another cell. So when we do have a pair of chromosome, usually what happens, we have a dominant gene and a recessive gene. Only the dominant genes are going to be expressed, not the recessive genes. So in case of a monoploid, since there is no other chromosomes for the regulation, most of the recessive genes which are deleterious in nature will start expressing. So as a result, so monoploid is going to be very difficult, and this is not at all possible in animal world, but in a plant world, yes, we can still get recessive characters can be expressed. So we can, that is what is known as an anther culture. Probably you have discussed all those things in detail in case of a plant genetics. But now my aim in this year to make sure that how colchicine interferes, and because of the non-disjunction there is an unequal distribution of these chromosomes. So now in the normal we all get n gametes, but in case of an abnormal, so we have n minus 1 and n plus 1. Suppose if you do take this variety n plus 1 and fuses with another n plus 1 gamete. So then what happens? Now this is an n plus 1. It is as good as a 2n status. Then another n plus 1, again a 2n. When they fuse together, now then what happens? We are going to get a 4n variety. So the chromosomal number is going to be doubled in case of a 4n. So suppose here if you do take the overall gene is 100%. Now in case of a 4n, that 100% gene expression will become 200% gene expressions. So as a result, the dosage of genetic material doubled. So the entire process, so suppose if an onion, if you do take of a particular size, so of 2n, and if you do take a 4n onion, the onion size increases from 4n to 8n. So now it still increases, but the other phenotypic characters will remain the same. But the plants are bigger, the flowers are bigger, the fruits are bigger. Now, so a small onion to a bigger onion is an advantage for us. So that is the application what we are talking about, the somatic cell fusion. Now the interesting one is going to be the triploid, because I told you that seedless variety. So triploids are always sterile. You are going to get a plantlet in case of a triploid, but this triploid will not provide any flower or any seeds because of several reasons. I will tell you that. So in case of a triploid, so we create a gamete like this, an n gamete and n plus 1 gamete. Suppose if there is a fusion between this gamete and the n plus 1 gamete, now what happens? So now here this is a 2n state and this is a n state. Because of the fusion, so we are going to get a 3n, a triploid state. So in case of a triploid state, so in case of a diploid, we have a pair of chromosome. Whereas in case of a triploid, instead of a pair, we are going to get 3 copies. Assuming a plant has got, for example, a plant has got 4 chromosomes. So now each chromosome, it will be a pair. But because of this triploid, so instead of 2 copies, we have 3 copies. So now when it comes to the 3 copies, now these 3 copies is unable to enter into meiotic cell division, because in case of a meiotic cell division, reduction has to take place. If it is an even number, for example, if it is 4n, so because of the reduction we are going to get a 2n. Or if it is a 8n, now the gametes is going to be 4n. But if we do have a 3n, so we cannot have a 1.5 gamete. So with a 1.5 number, so as a result, so during the meiotic cell division, there won't be a proper segregation or a disjunction, because we have 3 copies. There is no way 3 copies can be equally distributed. As a result, triploids are always sterile. So we are not going to produce any seeds in this one. So if you want to have a seedless variety, either a grape or a watermelon, all we have to do is that you do take a normal gamete. Then because with the colchicine we try to get an n+1 variety, n+1 gamete. When you fuse this gamete with n+1, we are going to get a triploid. So you do get a plant, they do get a flowers, but their seeds are of insignificance. So that is the strategy what they have used, so in order to develop a seedless variety. Now you get, it's a very smart. All it does is that colchicine will take care of the whole process. So now what happens is that here the mistake do happen in the second division. What happens if, because of the colchicine, if the disjunction is not proper, if there is a non-disjunction in the first division, what happens? The 2 daughter cells, this daughter cell will not get any chromosome, but this daughter cell will get a pair of chromosome. Alright. Now when you subject this to the second division, now there won't be any chromosome, so we are going to get 2n-1, and this will undergo a normal segregation. We get n+1 and then n+1. So now you take n+1 and then fuse with this one, we get triploid, or you take n+1 and n+1, we are going to get a 4n. So now you just got an idea about how with the help of a colchicine, so we can make a 2n to 4n, 4n to 6n, so on. Now you got an idea. So now triplets are the strategies they have used in order to get the seedless variety. So now this is a part of plant breeding, but similar ploidy is not possible in the animal world, because so they have a very strict regulation in which you cannot make a superman or a superwoman, because this type of manipulation is definitely not possible in the animal world, but it is easy to perform all kind of manipulations in the plant breeding. Now the next one, I think we will discuss about what we call this as an Allo polyploidy. Now I will tell you the advantages of an Allo polyploidy. So there is a famous experimental design by Karpachenko. Alright. So now what he has done is that, so he has taken 2 closely related species. So now this is a Raphanus. It is commonly called as a radish. Alright. Now the 2n number is 18 chromosome. It is a 2n is 18 chromosome. Then he has taken in a very related species, Brassica, also called as a mustard. Now that 2n number is also 18. Suppose during the meiotic cell division, the n now is equal to 9, and even in case of a Brassica, the n is equal to 9. Karpachenko was got the idea stating that in case of a radish, the useful product is the root, because the roots are consumed. In case of a Brassica, roots are not important, but the mustard seeds are very important, or the shoot pot is very important. So since these things are all closely related species, he thought, so we can generate an N gamete from the radish, which has got 9 chromosome, and also an N gamete from the Brassica, which is also a 9 chromosome. can we fuse because of the somatic cell fusion? So he got a F1 hybrid. So, but these N9 chromosome and these 9 chromosomes, they are not homologous chromosomes, basically because this chromosome set belongs to radish and this chromosomal set belongs to brassica. So during the meiotic cell division, there is no way we are going to have a homologous pairing. Even though they have 9 here and 9 here, since they are not homologous in nature, so there won't be any pairing takes place during the meiotic cell division. Now this really tells us that in case of a meiotic cell division, homologous pairing is very, very important. As a result, now the F1 hybrid are sterile, basically because there won't be any homologous pairing and they are not going to produce any seeds or any variety. During his experiments, during what happens is the F1 hybrid, a particular branch, they grow and develop flowers, and it was really, really surprising. So he took this particular branch and tried to analyze. So what happens is that, so now here this N and this N is becoming a 2N, but even though there is a 2N, because the homologous pairing is not possible. Now this 2N, because of an ploidy, Allo polyploidy, 2N all of a sudden becomes 4N. So now this 2N, so now we have the 18 chromosome. So in this 18 chromosome, 9 from Raphanus and 9 from Brassica. Now this 18 chromosome, because of a Allo doubling, so it become 4N. So now we have 18 plus 18, so 4N, we have 36. So now once we have 36 chromosome, so this 9 is doubled and this 9 is doubled. So now we have 18 of Raphanus and 18 of Brassica. Now what happens, this 4N, so now we have 18 from this, 18 from that. So now subsequently what happens, the homologous pairing is possible. So in the F1 generation, homologous pairing is not possible, but this 2N becomes 4N. So now we have 18 and then we have another 18. So now then once we do have 2N is transformed into a 4N. Now this 18 will find an additional 18 chromosome, so that this 18 can homologous pair with the another 18. So now this 18, this 2N gamete will become a 4N. So then what happens? So initially 2N, so we have a N, we have a fusion F1 hybrid or highly sterile. Now suppose if you can make this F1 into a F2 with a ploidy, so what happens, like now 18, we get additional 18. Now this F2 generation is fertile because they can undergo a normal meiotic cell division. So now we are going to get the new plantlets. So now from here, so we are going to get a 4N. So now this particular plant species can be cultivated. Okay, now, so he called this new plant species, now the Raphanus and the Brassica, he called this as a Raphanobrassica. Virtually, Carpenchico has created an evolutionary process in the laboratory and created a new species. Okay, his idea was to get the root of radish and the shoot of a brassica. Unfortunately, his new plant species has got the root of brassica and the shoot of Raphanus. So both of them are going to be useless. We don't consume the leaf of a radish, and we are not interested in the root of a brassica. So his idea was to get the other way around, but the experiment did not go the way he planned. But still, this is a milestone experiment principally because this Raphanobrassica is a fusion of this genetic material and this genetic material. In the F1 generation, they were not reproducible. In other words, they are not fertile. But subsequently, the F2 generation, because of the ploidy, so we are going to get a viable one. So this is the Allo Pollyploidy, where we can get a new species. Okay, now when you look into the cultivated wheat plant, okay, now currently available the wheat plant is called Triticum aestivum. Okay, so we always assume that this is a diploid with 2n, but when you do the chromosomal analysis, it is usually the Triticum species, so they are diploid or 2n, but the one what we are cultivating currently is 6n. Now they are wondering how we have Triticum aestivum with a 6n, probably because this is a somatic fusion of 3 different variety. Each one has got an advantageous trait, so we want to get the beneficial of all these advantageous trait, and we have included that in Triticum aestivum, which is a 6n. So now, because of continuous cultivation, different people, different farmers in the different part of the world, I think they have in a classical plant breeding. So now the initial one is going to be the Triticum monococcum, assuming that the trait has got an AA, this is the 2n status. Another one is going to be the Triticum cerci. So this has got one beneficial trait and this has got another beneficial trait. So now like this there is a somatic fusion. So now we are going to get the gamete, N gamete is A, the N gamete is B. So now we fuse them. So we are going to get the chromosome from this, but they are not homologous. Now we have an AB trait. F1 generation like this, they are very sterile, but if you can make them into a tetraploid, then it is going to be viable. Now this AB in the similar fashion is transformed into 4n, that is AA and BB. So now we have a pair of chromosomes which is possible to do the meiotic cell division. Now that new species is called Triticum turgidum. So now this particular plant was cultivated for centuries together, and later this particular hybrid variety fuses with another diploid variety, Triticum tauschii. So now what happens, the gamete of this is going to be the AB. So now this is 2n. So now we have, this is a diploid, the gamete of that n is D. So now we make a somatic fusion AB and D. Now this ABD, you think we have 3 copies of each chromosome, and again they are not viable in the first generation. Subsequently in the so now this triploid has become a hexaploid, AA, BB and a DD. So now that is what is called as a Triticum aestivum. Alright. So now how they found out that this particular one is a hexaploid? Basically because the Triticum species, so when it comes to the diploid, so now if you try to analyze, that is through the bioinformatics, the sequence analysis, now this particular sequence has got similarities. So now part of it has similar to this species and some is similar to this and some is similar to this. So now they have concluded that for sure Triticum aestivum is a fusion of 3 different varieties over a period of time, that is using the classical breeding. So now this is, so you can get a very good variety of agricultural importance now by this process what is known as a Allo Pollyploidy or a Pollyploidy. In a natural evolution, during a new species is going to be generated or born in the forest, they all go through this ploidy process of Allo Pollyploidy or Auto Pollyploidy. So this is about the somatic cell fusion in which new traits are going to be generated. But the important component is going to be the protoplast. You know how to generate a protoplast in the laboratory conditions, in fact. I think we will end this particular portions now.